You just got your first dose of the COVID vaccine and are really excited. In this video, we will show you exactly what is going on inside your body after you get the vaccine. The cells in your body basically function as large construction sites. Like a construction worker using a blueprint to build a house, your cells use mRNA as instructions to synthesize and assemble new proteins. We can also use our body's construction process to protect against those trying to break into our cells, functioning like a security system, using mRNA vaccine technology. In this case, our mRNA blueprint will be for the spike protein found on the COVID virus. The spike protein is what the virus uses to break into our cells. Translation occurs in the cytoplasm of your cells. The first step before we can actually build anything is to charge the tRNAs with amino acids, since we need amino acids to construct the polypeptide. Amino acids attach to the 3' end of the tRNA to form a charged tRNA. In our analogy, amino acids would be the materials our house is constructed out of, such as the wood, foundation, etc. Ribosomes are the main builder in the amino acid construction process. They are mainly composed of ribosomal RNA and protein. Our mRNA blueprint is not used in full. In eukaryotic organisms like yourself, there are areas in the 5' and 3' untranslated region that help facilitate the binding of the ribosome. Proteins on the 5' and 3' untranslated end help position the ribosome onto the mRNA at the AUG or start codon, the first codon that is translated. During elongation, codons in the mRNA base pair with anticodons on the charged tRNAs. The amino acid is held by the ribosome, and as more charged tRNAs come in, the ribosome assembles the amino acids together to form a protein. This process continues until a stop codon is read. After this, translation stops, and the completed spike protein is released from the ribosome, which can then train your immune system to attack it. That way, if it ever encounters the spike protein found on the actual virus, it will be prepared. Here is the gene sequence of the actual coding strand of the COVID virus found on PubMed. This is the original DNA and mRNA sequence, and this is the protein it produces. Synonymous mutations are substitutions of nucleotides that will still produce the same amino acid as the original codon. We would not need to make changes to the mRNA vaccine to account for this change because the amino acid sequence of the spike protein remains unchanged and your body will be able to recognize it. Non-synonymous mutations are substitutions of nucleotides that code for a different amino acid. A change of AGC to AGG produces a different amino acid than the original sequence. This will alter the spike protein, and we would need to take these changes into consideration because your immune system may not be able to recognize the mutated spike protein. To account for these changes, I would develop booster vaccines with the new mutated mRNA sequences so that your body can manufacture these new spike proteins and train itself to fight against them.